Okay, I think we can just get started. People are just trickling in now. So, yeah. Hey everyone, welcome to our speaker event with Dr. Lisa Freed, hosted by HS Robotics team Inca Medal 5773 and the Inspire Speaker Series Club at American High School. My name is Anuj Naik and I'm an outreach captain at FTC Robotics team 5773. And before we begin, we'd like to introduce our speaker and explain how this event will work, which Krish, the president of the Inspire Speaker Series, will start with.
Hi, everyone. I'm sure many of you already know this, but Dr. Lisa Freed is an MIT research scientist with 137 publications in the field of bioengineering. As a researcher, Dr. Freed specializes in the intersection of biology and robotics by working on groundbreaking prosthetics and human motion staples, regenerative medicine, and tissue engineering. At MIT, she is currently working on providing amputees prosthetic sensory motion and control. Through her role at NASA, Dr. Freed has contributed to the biotech facility on the Mir space station as well.
Thank you so much for coming today, Dr. Freed.
The way this event will work is that we'll begin with a 20 to 30 minute presentation from Dr. Freed on her work and career tips, and then we'll use the remaining time for an audience Q&A. Dr. Freed, would you like to begin?
Sure, thank you. Thank you very, very much for inviting me, Anuj and Maithili, and I'm happy to be here. I gave a title for my talk today of Biomedical Engineering and Bionics, and I'll just jump right in. When I started to think about this talk, I started with some thoughts back to when I was in high school and some fun facts. And I thought I'd just explain how I got to doing what I'm doing now. So all I could think of is when I was in high school, things were really, really different, and not just because of the pandemic or anything. It was just like a different whole time. Everything looked different. When we were, I know one of or more of you are applying to college right now, but that was different. TVs looked different. Toys look different. When I was in high school, this show came on about the bionic man and the bionic woman. That was before Luke Skywalker in Star Wars. And I was very intrigued by this show because she had 2 bionic legs, a bionic arm, and a bionic ear. And every time she needed to have any of these things fixed, she got sort of plugged into a station, and that's how it went. Anyway, after high school, let's say I had a lot more school for about 10 years and got several degrees, and basically took a lot of different opportunities as they came up, including after school, travel overseas, and then I became a lecturer in anatomy at Harvard Medical School while I was doing a postdoc, and then I've been a research scientist in a number of different places until the last 4 years when I came and settled upon the MIT Media Lab. And it was suggested that I ask several questions maybe to pose to the people, and so here they are, and I think Anuj is going to collect answers. They're sort of more like themes than questions for this talk. So first one, what is a bioreactor? The second one, what's a biomaterial? And these are all just parts of the field of biomedical engineering. What is tissue engineering? Then what is biomechatronics, which is the group name for where I'm working now? And finally, what is regenerative medicine? These are things I'd like you to think about. And when you see stuff in the next several slides going to the end of the talk, there'll be these kinds of ideas coming up. And a lot of these fields started pretty recently, actually, and a lot of progress has been made, and there's a lot more progress still yet to be made. So there's a wide open space for everybody. Okay, so then I had a request to talk about some work I was asked to do for NASA. There's actually a slight typo somewhere. It was not on the International Space Station, it was before then. was back at the last part of the last century in the 1990s, when there was the space shuttle, of course, but it was docking with the Russian space station Mir, and it was called the Mir shuttle program. And at that time, I was working on tissue engineering of cartilage, and I'll talk about that for a couple of slides. But basically, the reason I got to fly a space experiment for 4 months on this Mir space station that was delivered by the shuttle was because we had an experiment that interested NASA. They had been growing kind of crystals in microgravity, in the microgravity environment of space, but we were able to grow tissues in a kind of a simulated microgravity environment where they kind of floated around. So the way that we did it very basically was we took cartilage cells from actually a cow and we put them on what we call scaffolds, like the scaffolding you put around something to give it a structural support. And then we would put them in a vessel that was a tissue culture bioreactor. And there was one that was growing happily in Houston, Texas on Earth. And then there was one that was transported up into microgravity. And then because of a bunch of circumstances, that the experiment lasted a lot longer than it was originally planned to last. And it came down after another 4 months in space down to Edwards Air Force Base near where you guys are, and traipsed back across the country. And while it was in space, I should just back up a second. This is a picture of what it looked like, and these were schematics. But here there were little cartilage pieces that we had engineered that were sort of floating there. And this was the flight pilot who had to keep feeding it because the red material inside there is fluid full of nutrients, and this middle cylinder helps get oxygen in and out of it. So it's like an environment, but they were floating there for a while. Then I collected the samples and the team digested them down backwards so that they became the cells again. And then this shows that they had survived the whole spaceflight experiment, and we had the control experiment done on the ground and in space versus ground. There was only one bioreactor in each place, and so it was hard to know what would happen if we had more time points or some other things we could have done. But we were able to publish the papers, and it got into the NASA documents, and it was a really exciting experiment. And not only that, it got us a lot of funding. These were different NASA grants that I had over the years. And instead of only just doing spaceflight experiments, they had what they call simulated microgravity at 1G at Earth. This is what a simplified bioreactor that's less automated would look like. And there were the pieces, and it turned out that these are rotating bioreactors. So they rotate as a solid body around and around. And if you guys are not seeing the video, let me know, please. But you can adjust the rotation speed so that a big piece of cartilage can sort of be suspended in sort of buoyant, neutral buoyant state, or you can actually use this for all kinds of cells and tissues that are even lighter, and you have a three-dimensional tissue culture environment that's kind of unique and interesting. And so we did a lot of studies in this kind of a model system. And also, as part of all these research grants, I got to do a lot of outreach. They lent me their shuttle model and their astronaut costume, and I could go to different schools. This one was just shortly after the Challenger had an accident, so that was an important time to go around talking about NASA back then. So that's just a little introduction to the work I did with NASA, but went on for like around 10 years. Let's see. So more generally, tissue engineering has extended quite a lot since it was first developed back in the late 1990s, where you can put just about any cell, which you can grow up in a culture on a sort of a flat dish, onto just about any scaffold, which is widely varying and depends only on the limitations of a person's imagination. These are just some pictures. I'll show you some more. And then you can try to make at at just about any scale, going from nanometers to like centimeters and tens of centimeters, tissue pieces, including the cartilage, which, as people probably know, is the tough material that covers the surfaces of your bones to stop the bones from wearing. We worked a long time on this application, which is a hip repair. And then we also worked for a long time on an application like this, which would be a cardiac kind of a patch. So the lower one would be if a person had arthritis, the upper one if they suffered from a heart attack. And these tissues and some other ones, like the nerve tissues we're working on now, are usually picked because they don't self-repair by themselves. So if you can make a patch, that can be helpful to people that had injuries and various other pathologies. So one example that we were working on was in an animal model. This is the knee of a rabbit. And the surgeon created a gaping hole that would be too big to self-repair. And then this is one of the material scaffolds that we developed early on. It's just like a fuzzy felt, but it's a special degradable, compatible material that it just serves as a scaffolding, just like when you're building a building, then you take it down when it's all built, because sometimes these materials can get in the way and sometimes you do need them to stay, but other times you don't want them to stay. So you can tune that property. These were some what are called mesenchymal stem cells, which we could take from this same rabbit's own hip to what we call seed onto the scaffolds and grow a kind of an implant. And this was the kind of an implant we first put in after a 6-week time point. You could see it because it looks different from the rest of it. And this was a bone support matrix. And then at the next time point, the final time point after 6 months, you can see how the cartilage, which is the dark purple layer over here. Whoopsie, I'm sorry. Let me just back up for a second. This dark purple layer is the very surface. It's usually very thin, like it is right here. And then other tissues had regenerated underneath. And that way you can get quite a good repair. There are a lot of different markers that you can use to see how well this is happening. So that was an example for tissue engineered cartilage. And then I jumped the slide to a completely different kind of a scaffold that we also made by microfabrication, actually, that was for heart repair. And this one, well, the concept was it was going to look like an accordion. And that was so that it could stretch back and forth like the heart does. This isn't a video, but this was a piece of rubber in a stretching device, and it was called a BioRubber Scaffold. It was really different from the kind you need on your bones and your joints. So if you see this one with heart cells instead of with the other type of cells, like the normal heart, where you usually get elongated cells that can contract, we got some elongated cells inside the scaffold. So these are fake colors that you put on this kind of a microscope image. You colorize them yourself, but you can distinguish the scaffold material, which is gray over here, from the cellular material that grew on it after it was seeded and cultured there, which is green. And if you see this video, I'll play it twice. Can you make out that the cells are actually contracting in there? And they're just living in this case in a petri dish. So it was quite cool to see them contracting like that. And the only problem was that these accordions weren't going in and out because they were still too stiff, and they were still pretty thick. Because now over years, these are only 50 micrometers thick, so they're about the size of a human hair, but they're still they were not as they were stronger than the cell's contractile force was. So this was a study that was done, and then we were carrying that even a little further with multi-layered scaffolds, because one thing that the heart application needs is, of course, a blood supply. And in the heart, every single heart cell is like very, very closely placed to a vascular cell of the blood vessel system. So we made a microfabricated device out of this same biorubber material, a degradable material, which we could have microchannels. It looks kind of like a boat, but then you get to the central part where these channels lead in, and then this is layer by layer by layer. There was a kind of a porous layer in between with that was like a membrane, and then there were several layers of a scaffold above. We could seed the vascular cells first at the bottom layer, and then we could seed heart cells second. We could grow them by pushing through culture medium this way. Then we could take it and put it in a rat heart. So these kinds of tissue engineering experiments are basically only limited by the ideas that you have as a researcher, and all kinds of tissues are being generated this way in the laboratory setting, and some of them are actually being implanted and used to for all kinds of medical purposes. Others are being used for diagnostic purposes, to study drugs so that you save animals' lives, or to study the immune system, all kinds of applications. So these are kinds of tissue engineering work. But now I'm going to switch gears a little bit into, it was mentioned that I was asked at doing all this tissue engineering to advise for FLL, the first LEGO League, and back in 2010, this was their 2010 game board. So 4 of us went to New Hampshire and met with people that design these Lego pieces. And this is a game board that's used with the Mindstorm robot, the Lego Mindstorm robot. Maybe some of you did that Lego League before FTC. And so there was a game, and it was designed, and this was our heart patch application. The people made a heart, and the team members had to put the patch on the heart. And then this was a different bone repair application where the leg was broken. There's a foot here, and you had to put a patch on the leg, and that allowed the foot to kick the ball into the goal. And then this was a nice, the robot had to bring an engineer together with a doctor and a patient all in a certain spot on the board. So there could be a healthy collaboration between the doctors and the engineers. And then the last one was basically my favorite one. At the end, when you had done all the things you were going to do, in the center of the board, there was a patent application. And if you were the first to come and grab it, then you got the patent, and it was all scored up, and it was a lot of fun. And it was the first time I ever even thought about robotics, and I thought it was very cool. So the whole experience was very cool. And then I mentored a couple of teams, and it was fun. So that led to what I'm doing right now. This is the team I'm working with right now, and I'll tell you more about some of them. And we're working in the MIT Media Lab Biomechatronics group. And this is the leader of the team for whom I work, and some of the people who are in my immediate group or were from all over the place and with all kinds of backgrounds. These 2 are medical doctors, and some other people are programmers and mechatronics people and mechanical engineers and embedded software and embedded electronics, and all kinds of different backgrounds, because this is very interdisciplinary work. And here are a couple of pictures from our lab. This one is inside the lab. There's somebody is back there having electrodes put on her leg, and then these are summer interns. He's actually a student with us now, and we're trying to make it work. And then we went out and ate. So we have a lot of clinical research subjects coming in and out and donating their time, well, for a little bit of reimbursement. And without them, we couldn't get any of this done. So I'll talk about that pretty much for the rest of the time. Here's the statement of what we're trying to do. So we seek to provide these people that are living with amputations better capabilities for sensory perception, motor control, prosthetic joint stability, terrain adaptation. We do this clinical research using a reconstructive surgery technique and especially adapted prosthesis. And this work can potentially impact the quality of life of 2 million Americans who are living with limb loss. And this number is only growing and is expected to increase dramatically even by the next couple of decades. And this is the leader of our group, and his name is Hugh Herr, Professor Hugh Herr. And when he was a teenager, he had a terrible accident where he got frostbite on his legs, and they had to be amputated below the knee. And yet he was a rock climber, and he wanted to rock climb, so he began to build his own prosthetic legs, and he ended up able to climb very, very well with his prosthetic legs. But then he went on to get a PhD in biophysics and learn how to make better and different prosthetics that had ever been made before. And he has a TED Talk, and you can see he had a company, which isn't his company anymore, but which is still making one of the only available powered prostheses that you can get for an ankle robot. And so there's a brief video clip. You can see him wearing the prostheses he's designed and talking. I'm not sure exactly what he said, but he demonstrates that, he's able to move around quite well on them. And they're intrinsically controlled. There isn't any direct neural connection between what he wants them to do and what they're doing. However, they are powered, so they can give him like extra power when he pushes off from his calves and whatnot. So how does this work? How do we get this human device communication? And what is this special reconstructive surgery anyway? This is a schematic. There are going to be more. So if you don't catch it the first time around, I'll explain it a bunch of times. How do you get a signal down from the brain to the bionic prosthesis and get a return signal back so that the person can control the prosthesis with their own mind and also understand where it is in space? For this, a surgical reconstruction was sort of invented, which is called the agonist-antagonist myoneural interface, which we call the AMI for short. And it's shown here, but I'm going to go to the next slide to show you a little better how it works. So if you have no amputation and all your anatomical parts are intact, your muscle pairs are arranged around the joints, like your arm, the biceps and the triceps, around your ankle. You've got your calf muscles, and then you've got some other muscles in the front that people don't always know the name of that are acting opposite to one another. When one muscle contracts and tenses up, the other one, opposite one, stretches. That's called the antagonistic action. However, when you have to have a amputation, those normal anatomical muscle pairs are severed instead. And for example, the calf muscles sort of wrapped around and sewed together to create a nice place to push on the prosthesis. How then can we preserve sensory perception? Well, the theory what we're investigating is that with the AMI amputation, we preserve the normal physiological muscle sensors. And in the muscle, if your muscle is relaxed, you have something called a spindle fiber that's relaxed. When you contract it, the amount of basal signaling that's coming out of this sensor is reduced. And when it's stretched, the amount of signaling, the frequency, is increased. And again, there are force sensors at the end of your muscles where the muscle attaches to the bone and the tendons. There are another type called the Golgi tendon organ, which is sensing the force. So with the operation that I'm going to show you in a video, starting with this scene, you'll imagine how there's a way that we can try to preserve these physiological muscle sensors instead of just severing them and keeping all the muscles that are left sort of isometrically positioned, because that way you lose your sensation. So here's a movie of the AMI surgery during an amputation. You can harvest tissues. In this case, you're going to harvest a tendon and its sheath in 2 places. You're going to attach it onto the remaining bone so that you can reconstruct the muscle pairs to go around. And then they normally signal up and down the leg to the person's brain. And if connected to a prosthesis, the person can imagine, perceive what the prosthesis is actually doing, because the muscles that you've reconstructed are actually sensing and commanding this prosthesis the way that you want it to. So it gives you a non-intrinsic method to control this ankle foot. And that's what's being investigated in the study. So the biophysical design concept is shown over here. Here would be the 2 muscles sort of in their most simplified format, A and B. You get a nerve signal into muscle A and it contracts down. And since it's directly connected through this tendon to muscle B, the fact that one is contracting stretches the other. You get a second signal out, a change in that signal out, and that'll go back to the brain. Here it's written out in the patent application a little bit differently, and here it's a little bit more anatomically arranged like you saw. So we'll call one the agonist muscle, the second the antagonist muscle, and then in between is that pulley where they took the tendon in its sheath and they reconstructed the muscles through it. So the vision is that for each degree of freedom of your prosthesis, you need 1 AMI muscle pair. These are also called the AMI constructs. So if you want to dorsiflex or plantarflex your feet, which are what are shown in the cartoons, you need 1 degree of freedom for that. And that's your ankle joint up and down. Just think of pushing on the gas pedal. But your ankle has 2 degrees of freedom because it also turns from side to side, the eversion and inversion. So you need a second AMI construct pair. And then you get what's also anatomically called the subtalar joint. You can evert and invert your ankle. And you can imagine there are 17 degrees of freedom in your hands. So you would have to modify this for the more degrees of freedom that you need. Or if you lost your knee and your ankle joint, then you would need another AMI pair. So you can extrapolate from there. So now let's get to some of the work that was done. This was done actually right before when I was just joining the lab. And this is one of the patients with an amputation on his left leg. He was the first patient we had. And these are wireless EMG transmitters that are sitting on his AMI muscles on the skin. And here will be the muscle signals. They're called electromyography EMG. And he's sitting there and he's asked to mirror, meaning he volitionally moves his amputated limb in the same way in a mirroring pattern to what he's doing with his biologically intact limb. And you can see the way the signals come out. Now he's going to circle the other way. He's like drawing circles with his feet. And this is the kind of data sets that we have to analyze. So the next several slides are like this one. There's no prosthesis attached, and they're called free space experiments because there's also no mechanical resistance operating here against his movements of his amputated leg. So how can we build now? Because I'll just back up for 1 second. I meant to say that these devices, they work very well, but they're very big, and they would be very hard to put in a prosthetic socket. They're about a centimeter high and they're bulky. So one of the things we recently worked on was, here would be your skin layer, this yellow, and your socket would be the green layer, and it's usually made out of silicone rubber. We would like to have in-socket electrodes that are comfortable to wear, easy to manufacture, affordable and reusable, and compatible with a person's own prosthetic liner and socket and prosthesis. Because each person with an amputation has a whole bunch of equipment, and they don't want to have a new set just because they want to be able to use electromyography to electromyographic signals to control a prosthesis. So those
Dr.
three.
Fried, someone
Yes.
actually had a question relating to one of the slides before.
Oh, sure.
So they asked, I think this was for the slide just before, does this operation assume that the calf muscle remains intact? Are most below the knee amputations done even if only ankle and foot is injured?
The calf muscle is not intact because the calf muscle attaches to the heel. So the calf muscle had to be severed and reconstructed during this. So the calf muscle, which is called this one, the gastrocnemius, is shown. It's not, it's functioning, but it's not intact. If by intact you mean whole, because normally the calf muscle starts up way, it's a biarticular muscle which attaches to the femur and then attaches on the top of the tibia, and then it goes all the way down to the Achilles tendon, which attaches to the heel. I missed, sorry, the second part of the question though, but it's still working. We're going to use the calf muscle. What was the second part, Anuj?
They asked, are most below the knee amputations done even if only the ankle and foot is injured?
Yes, usually you have to be able to walk on something. So if you can no longer walk on your foot, the idea is to create, and it can be lower down or higher up, but depending where the injury is and what it's from, it's usually done for a trauma or sometimes there's a tumor, or sometimes there's a congenital problem that you're born with your foot, like a club foot. There are lots of different reasons. Sometimes the blood supply is a problem in diabetics, but usually you need something to walk on. So yeah, they save the knee, and then usually they're done in the middle of the calf, like was shown. Does that answer?
Yeah, I believe so. Thank you.
Okay, sure. So I'm going to go to the next slide because I was up to it anyway. We designed, or actually Sung Ho Yang and his sub-team designed what's called a slip electrode, like a slip that you wear and it slips under the silicone prosthetic liner. Oopsie, excuse me, that was too fast. The electrodes are here, and they're in between the socket and the residual limb, which is what we call the rest of the limb that's left after the amputation. The electrodes themselves are very flexible, and they have a long cable that comes out and attaches to embedded electronic wearable surface EMG acquisition and processing system. They're made out of Kapton, which is polyimide polymer film that's only about 100 micrometers thick, and they're gold-plated copper film underneath this insulating substrate. And so that's the concept of them, and this is how they actually look. This is a different person with an amputation, and she has, you can see 3 on this side, and there's one on the other side. And for comparison, these are standard. These are called Exg electrodes. Some of you might know them because you can put them on your scalp for the brain studies, and you can put them on your chest for the heart studies, and we use them all the time normally. But again, they need a sort of a thick preamplifier here and then a shielded wire, and they are not comfortable to wear inside of a socket. And also, you can't get as many of them inside. So here we usually use 4 pairs of bipolar electrodes, but we can actually accommodate 16 pairs. And here's a scale bar there, about 1 centimeter each, coated with gold, and the intra-electrode distance is about 1.5 centimeters, 16 millimeters. Then you've got this long cable that you can thread up through the liner and the socket as is shown here. And so here was this isn't a movie, but it's a picture of this woman doing about the same thing as you saw before, only this time her amputated leg is got the electrodes under a socket and on a liner, and on the top, which is also shown here, is this very cool system that you can walk around in, not tethered, to read out the EMG signals and to log them wirelessly, so that they can be collected and used to control a prosthesis. So here's an example representative, and this is in free space. So here this is the name of the calf muscle. This is the name of the muscle that opposes it, the antagonistic muscle pair, blue and yellow, for the ankle joint to move the foot up and down. Then the other pair is, these are the names of those muscles that are on either side of the ankle, moving it side to side. Those are green and red. And what she was asked to do, and this is the EMG signal in a time domain so that it can be used in a time domain to control the robotics, is to, again, make circles, but then to rest in between. And I don't expect you guys to be able to see, and it's not even clear yet, except that there is very good signal resolution, even though it's being worn inside the socket. And there are different things happening with the different muscle pairs, and that when there's rest, there's not a very lot of baseline going on. But I'm going to move on because I'm going a little slower than I had expected. And so how we use this is the last free space experiment slide, is there have been many different control algorithms, and I'm not going to dwell on them in this talk because it was broader than just that. But in this person's, his name is Jun Qing, he's in the middle of working on it. He collects a training data set as she just moves her foot back and forth in a kind of a random way, in a way that maybe it can be tuned even at home, even without a person coming in, if you change electrode positions. And then the screen displays, it's a graphical user interface, and it displays a series of targeted movements. So if the rest position is in the center of this screen, marked as sort of in red, the cursor will go someplace, and her task will be to move her amputated leg muscles so that that foot would point at that spot. And then she's supposed to stay there and hold it, stiffen her muscles, and then go back to the rest position, repeat for a series of targets. And for that, we are collecting clinical trial outcome measures, because you always have to have your plans for human clinical trials beforehand and then follow what you plan to do. So the outcome measures would be, how stable is her joint position control? Can she really hold these positions? And how economically can she move from position to position? I mean, how much wasted effort is there? So those data are being collected now as part of our clinical trial. But then you might ask, what happens when you put on the foot? And so there are a couple of ways to do this. This is actually the same subject, and this over here is a second subject, which you can tell. This is a robotic prosthetic ankle foot that has 2 degrees of freedom, and it attaches to a standard socket mount, and it's powered. It has this big battery, and it has belts and a lot of gear inside where you can assess how much torque is on the joints in either direction, and where you can understand how is it actually moving in response to her commands. And you can see the EMG data acquisition system up here, and then you can see it again over here. It's not, this is a regular outlet. This is an untethered system. What she's doing here is she's going to walk on a split belt treadmill. You can see it over here. So you've got force plates underneath, and they can take her regular biologically intact foot or her amputated foot and this prosthetics ground reactive force data from it. Then there are a couple of ways that we can look at the rest of the joints. And the joints are the ankle of the prosthetic, which has its own sensors. There's her own ankle joint. Then there are her own knee joints, and then the hip. We're looking at all those joints and how they're moving. We can either do them with a motion capture system, which are why there are these little balls. The little balls are reflective, and there are lights up above, and there are cameras that you can watch where the anatomical parts are and create kind of stick figures using this. But this is kind of takes a while to set up. So sometimes we do this, but sometimes we do this. And what we have over here, you can see nicely on his 2 knee joints. These are 2 degree of freedom goniometers, which measure your joint angles more directly. in 2 degrees of freedom and are also wireless. So here we have we can also put, instead of these reflective balls, we can put goniometers on the hips, the knees, and the unaffected ankle joint, and then get the data that way. So we can take usually 8 different EMG sensors and then 5 goniometers of data, and we can analyze the gait at level ground. So, and she does not have to wear the robotic ankle. She can wear a standard prosthesis as well, which she was doing here to test just the electrodes. So here is the gait cycle, where there are these different phases, and we need to use an intrinsic control algorithm for when the foot's on the ground, so that the person can use, does not need to have their own control. However, the reason that we want the EMG control is in case they want to position their foot in a different way, or if they want their reflexes to kick in, or if, for example, they're not paying attention to where they walk and they encounter an obstacle or something in their way. Then if you can directly connect a sensation up to the central nervous system, you'll have less chance of falling, or at least that's the goal, is that you can enable the EMG control during the swing phases of the cycle. So there's the swing phase, which it starts out over here, and then there are phases of the cycle where the algorithm is just going along as a state function. So that's how this work is being done right now. And this is how it looks right now. Here are the same 2 muscle pairings. This is the calf muscle and the front muscle. And you can see this is the what's happening with the robot. The robot is transmitting back these data, which are in sync with the EMG data that are happening for the time that they're on the ground and the time that the foot is off of the ground in the swing phase. So we're just starting to get these data, and it's very exciting. And not only we're working on level ground, but the people are climbing upstairs. And these are instrumented stairs so that you can collect data that way. And you can also collect the data from the robotic foot itself. And so the clinical trial outcome measures for this terrain navigation of the stepping stairs are what happens during the stair ascent. Some people tend to, like without amputations, anticipate a stair, and as you're setting your foot down, you dorsiflex your foot, meaning you sort of get your toe out of the way. And oppositely, as you're going down the stairs, you kind of anticipate the stair, but you don't even have to be thinking about this, and you plantar flex your foot. So the question is, once you have this AMI surgery, do you do those things that we do without amputations, or not? And so those are the things we're trying to compare when we compare people who have and who haven't had the reconstructive surgery. And the last part of our program is the least far along so far, but you might have noticed that so far there hasn't been any feedback in the control loop at all. And I see some people smiling, so that's what we're heading towards. And there has been some demonstration that you can have basically a bidirectional communication back from the prosthesis to the brain. But so far, everything we've been doing has been just motion control and motor control straight down through EMG. So what we're doing here is, for people that have had this operation, we can use these AMI muscles that are reconstructed as a sort of a bidirectional signaling hub. So if you are asked, the person sits where he cannot see the robotic foot. The robotic foot is not attached to him, and he only has the muscles in his amputated side, and he has his biologically intact side. And he's asked to push down as if he were pushing down the gas pedal, like say halfway to the floor, or all the way to the floor, or 75% of the way down. And he tries to do that, and then the robot reads back out how far he actually gets, and he can do it with mirroring or without mirroring of of the other foot. Depending on what the robot reads out, you can then put an electrical stimulation signal into the opposing muscle over here. And the way that happens right now, because there's nothing implantable that we can easily use, is through functional electrical stimulation by very, very fine wire electrodes. The picture on the far left is where the clinician, using aseptic technique and a very, very fine wire, he can instrument the muscles that are close to the skin and put very tiny fine wires in, which will be taken out at the end of the experimental session, so that depending on what the patient response is, you can stimulate and therefore close the loop back to the brain, like was in the original video that I showed at the very beginning of all of this. So our variable parameter is whether we have or we don't have FES, functional electrical stimulation feedback, or how does that work? And the clinical trial outcome measures are how well can you feel the position of your joint in space. Proprioception just means where is your foot in space. Does he know if he's pushing this halfway down, all the way down at all? And how well can he control the torque, which we can take from the prosthesis. So those are the kinds of things we do when we do human clinical trials. And with that, I'll wrap up the talk and get to the Q&A. I want to say huge thanks to a huge number of people. I didn't do most of this work. I was very fortunate to be involved in it and learned a lot and helped a lot. But these are really team projects. There have been a lot of really clever people that I've been fortunate to work with. We were really lucky to get funding too early on from NASA, then from three different institutes of the NIH. And the last part of the talk, the bulk of this talk, could never have been done at all without really loyal, dedicated, unstoppable clinical trial participants who spent lots of time with us, put up with a lot of breakdowns and delays of every possible type, even during the pandemic. So I thank you for all your attention, and I'd be thrilled to answer any questions you might have.
Yeah, so thank you for that great presentation. So now we're going to be moving to the Q&A section. So during the Q&A, everyone in the audience will be able to submit and upvote questions on an online Q&A software called Mentimeter, and the link for that has been sent in the chat. Our team will select questions from the audience and based on our own research and read them off as they come. And Krish, you can take it away with the first few questions.
Sure. A question we often see from students is that they typically want to ask the speaker how they themselves could pursue the field that the speaker is pursuing. So I sort of wanted to ask you that question as well. I know at our school, and especially at science fairs, there are lots of students who are already starting to look into biomechanical engineering. Do you have any suggestions for how students can get started with learning about bioengineering, or even getting started with biomechanical projects of their own?
Right. So I mean, there are the courses, and then there are usually lab projects in the courses that are. That's why I did it theme by theme by theme. I'm not quite sure I understood the question, Chris. I'm sorry. How do people get started?
Yeah, or if you had any recommendations for any students here who might be interested in pursuing some biomechanical projects of their own.
I mean, usually once you're in college, you can take on a research internship, and sometimes during the summers of high school, you can as well. And my recommendation is you go for whatever opportunities you can find, and you seek them out if they're in an area that you think interests you, and you read and read and read, because the more you read, the more you realize how you can communicate your ideas and your interests to people that might be able to help you. Because I was just following a lot of sort of lucky leads and see what's new in the field. Some of these things I've been talking about were relatively new. So what you're really interested in, if you find something you're passionate about, read as much as you can about it, and you find out who who wrote it, if it's a paper in a regular journal, and you send them an email or LinkedIn and you say, I'm a student at blah, blah, blah. I'm really interested in what you're doing. Do you have any more reading to suggest? Do you know any people that are looking for summer jobs for a person like me? If you get to college, the same thing. A lot of colleges have undergraduate research opportunities for people. Start. You might not like each one you try, but get your hands wet. I mean, the kinds of things you're doing with FTC, it's great. You learn teamwork, you learn how to communicate what you did. You learn how to explore your ideas and, move from one idea to the next, build on each other's ideas. The kinds of things that all of you are doing in that activity are a perfect example. Just if you're interested in the more bio side. I don't know what courses are offered at your school, or if there are any opportunities to do science fairs and things like that. Usually there are.
So essentially your advice would be to have students get as much practical experience as possible while
and read as much, and read and pay attention to courses and which ones to take. If there are choices, talk to people about where you have really good mentors and teachers. It's a combination. I mean, usually if you see something and try something, then you get interested in it. You start Googling if you're a curious mind. And then you can learn a lot online too. You don't only have to learn from the course material. But then I guess my advice is that if you see a good opportunity, or you even have a feeling that you think this might be interesting, or you hear a good talk and you think this person might be a great advisor, or they might know somebody who is, just try to pursue things, within the amount of time that you have, just to try things. Don't be shy to try, because if you don't try, you won't know. Maybe you hate biology and maybe you love it. Maybe you don't like programming, or you think you can't do it, and then you try it and somebody teaches you and you get all inspired and you learn something really hard. So that's my advice.
Got it. Thank you. And our final question for you is there something or someone that inspired you to get into bioengineering or tissue engineering?
I think I was always interested in it. Maybe that bionic movie. I don't know. I mean, I had a lot of wonderful mentors. They were listed on the last slide. Let's see, who are my mentors here? Somebody helped me with my PhD. Her name is Gordana. He helped me with my PhD. She helped me when I was learning microfab. A lot of people. I put my father and my mom down. My mom had a little dissecting kit. She let me dissect stuff in our spare room. So, my dad was a scientist. Sometimes parents have, or friends of your parents have, access points where you can find out things that are interesting to you. Yeah, mentoring is really important. Try to find mentors wherever you can.
Yeah, I think that's really solid advice. I mean, a lot of times students think about really learning, but not so much about finding people who can help you learn more. So I can definitely see how that would be useful.
Yeah, if you can. I mean, a lot of people really love helping other people. It makes them feel like they're, giving back. Somebody helped them, then they help somebody. It's fun to explain what you're doing. I have fun when I get to talk to people. If you're writing something and you want to find somebody to read it over before you turn it in, if it's important, listen to what they have to say. You don't have to take what they have to say and use it all. You don't have to rewrite what you were writing, but you can find out other people's points of view, and maybe you're stuck on something. Don't be hesitant to ask, because it's no harm in asking. You don't have to really do anything. You just try to find out. If you have an idea and you want to find out whether it's really good, and you find somebody that might be able to tell you whether they think it's worth pursuing or not. I mean, that's really important. A lot of times people have ideas and they're great and they don't test them out, and then they move on to the next idea whereas it was a really good idea. Or other people follow a dead end and they never bother to ask anybody if it's worth keeping working on, or whether you move on to something different. So, I say interact and don't be afraid to ask and try new things. I mean, you guys are all at the cusp of, seeing what you want to do. And I thought I was going to be a clinical doctor, and I didn't end up doing a lot of clinical medicine, only in the context of research. So you don't always end up doing what you think you're going to end up doing. Something else comes along and then you start doing that. Other people, they just go straight as an arrow and they know what they're going to do and they do it for their whole life. So it really depends on the person. But you can always try and change or try and expand. It's kind of like a bigger pizza.
Yeah, well, thank you so much for the advice.
Sure.
I think in the interest of time, we're gonna end our Q&A there.
Sure, thanks. Thanks for having me. It was a pleasure.
Thank you for that great presentation and Q&A. I'm sure it was as interesting and insightful for me as it was for everyone. Thank you very much for attending our event, everyone. Dr. Freed has numerous publications available online. Make sure to check those out, and the video we will post of this recorded session. Also, make sure to fill out our feedback form. We really appreciate any feedback you have for our event, as we always strive to improve in the future. Please make sure to check out our Facebook group, YouTube channel, and Instagram for more updates on future speaker events. Some helpful links have been posted in the chat.
Thank you for coming. Thank you for coming.
My pleasure. Thank you. Have a nice afternoon.
You too.